to use UCSF Chimera to set up ligand docking using AutoDock Vina. If you're not familiar with the docking, you can search for documentation. Just type docking and you can search AutoDock Vina, which is the actual software that runs the molecular docking. You will be taken to this website. You can read the original manuscript here, the paper. And in this tutorial, I'll show you how to set up your docking simulation. Okay, so first we'll start with a simple model, and this will be trypsin benzamidine complex. So I'll show you how to set up everything. So let's grab it from a PDB. Upon opening, we see a nice structure. We have to orient ourselves to find our ligand and prepare for docking. Now, typically what's needed for the docking simulation, you need your ligand and you need your receptor. In this case, our receptor will be the enzyme trypsin, and the ligand will be the benzamidine complex. And I'll show you how docking results can give you excellent results comparable to a solved crystal structure. So this is a crystal structure of this benzamidine trypsin complex. Okay, active site is here. Here is the binding site for this benzamidine. You can see it forms nice salt bridge at the end of catalytic pocket here with this aspartic 189 residue. Now you see here that Chimera tries to show you certain ligands. For example, this is the calcium ion complex here, but it's not needed for our simulation. So we have to clean this receptor model. We have to split this complex into ligand and receptor files, and then we can get ready with simulation. So let's do that. So first, I'm going to select the benzamidine residue. Okay, and I will remove all the other atoms. So let's invert the selection first. Invert all models, and let's delete all the atoms. Okay, so I'm left with the benzamidine residue. Okay, so let's save this as a PDB. Save PDB. Okay, and let's give it a name, ligand. Ligand in a PDB format, and we are going to save that. Perfect. Now we can close this file, close this session, and reopen the original complex model in order to extract receptor molecule. So I will close the session and will return again with the starting structure. Alright, so I'm loading back the structure. So this time we have to get rid of the benzamidine residue and additional residue which are non-proteinogenic, so no protein. The simplest way I think would be just to select structure and select protein. Perfect. Then we are going to invert selection, invert all models, and we are going to delete the atoms. Everything seems to look okay. And now what we can do, we can just save this as our receptor. Save PDB, and let's do receptor. Excellent. So now we have our two structures ready, so we can set up the simulation. First, we have to load both structures. So this one, let's see what we have here. We have our structure. So let me just rename this model. So I will rename this as receptor. Excellent. And now I have to load also the ligand file. Okay, so this is my ligand here and my receptor. So here you see the model panel, and it's shown that I have a receptor and a ligand. This is the ligand, this is the receptor. Everything is cleaned and ready to go. All right, so to launch the AutoDock Vina interface, we're going to go Tools, Structure, Binding Analysis, and AutoDock Vina. Okay, so this is the window. We have to specify our output file. So let's browse it and let's do docking. And you can notice that the file type is different. So this is a file format called PDBQT. You can read in the documentation how the format is made. Basically, this is a PDB file format plus charges that are applied on the atoms. Okay, so docking, and let's do benzamidine trypsin. Trypsin docking. Excellent. Once we set out the output location, we have to specify our receptor, which is the same name, and our ligand. Okay. Number 0 and number 1, as it's shown here in the active models. Okay, now we have to tell the software how to select the simulation box. In order to speed up the process, we know that catalytic site of trypsin is found here. Maybe I can show you that quickly by running a surface show. Oh, there is some warning. Don't mind that. And here is our binding active site and binding pocket here. So benzamidine should be docked right here. So we have to specify in Chimera what is the surge volume. So we are going to create a box, and for this reason, if you're on Mac, you have to change it to whatever works for you. I will do Control and button 2. And I can click that so I can resize. Upon clicking on the window, active window, it should start drawing your box. So again, Control and button 2. All right, so you see, with these lines, I'm selecting the active site where the docking will take place. Now, to resize the box, it's best to move it around and click on particular surface of this box, and you can extend it using the same pair of buttons. So in my case, this is Control and middle click. So I will Control middle click on this face, and I will extend this. Now, the higher, the bigger the box, the longer the simulation will be needed to run it. I see this box is totally too big, so I will just extend it. I know that my active site is somewhere here. This should be fine. Okay, if I'm happy with it, I can move to the next step. See, this is the center of the box, and this is the box size. So I can, let's say, change it now. Since I know my approximate location, I can change to 16. I can change that to 18, and this one to 16 again. Excellent. So for the following options, we can select that if we don't have hydrogens, Chimera can add automatically. So let's keep it by default. We have to create the charges. So Chimera will apply charges on particular atoms. We'll ignore waters, and everything else will be as default. Now for the ligands, the same is true. So we'll merge the charges and remove it from the polar hydrogens. So we'll leave everything at default. Now for the binding modes, you can go from 1 to 10. The number of binding modes tells you how many models will be produced. Let's keep it at 5, let's say. Exhaustiveness of search will be at 8, so this is very precise. And you can also specify the maximum energy difference between the two best results. You can keep this as a default, or you can change it, let's say, to 2 calories per mole. Now, for the executable location, you can install your Vina. You can download it and install it using website. Just go and download, or you can use the Opal service, which is provided for free. Now, this is a little bit, if you're planning to run serious simulations, I would prefer that you install it locally, and that's going to be just faster. I will keep it local. Let's see what happens here. Okay, so once everything is set, I will click OK, and I will wait for the results. So you can see here, Vina is running right now, and we have our results. So that was very fast. In this View Dog menu, you see that you have your scores. I refer you to the manual and the original paper to read a little bit about what it means. And now you can click through each proposed model, and you can see the difference in RMSD, root mean square deviation, how far your next model is from the first one. So of course this one has a RMSD of 0. The next one is kind of moved, and the RMSD is calculated. Okay, so now you can see here that it's pretty, this benzamidine sits pretty nicely in the active pocket. Okay, everything seems to be okay. And in the model panel, you will see that we have 5 models of this benzamidine trypsin docking in PDBQ format. So what we can do right now, we can remove the surface, okay, just to see how it looks like on the inside. And you can see that we have here a nice complex, and it looks very similar to the original one. So if you're curious how this works, what is the precision of this docking, we can compare that to the native structure and see similarities. But before we go into further analysis, I would like to show you how what are the cool features here in this dialog. So if you go, for example, to Movie, you can play through all the docked structures. So you can inspect them visually. Okay, and you can stop the movie just the same way. Also, what you can do, you can save your results here by going File and Save, or File, Save As. And then you can provide your name, and this will be in a doc format, so .pdbqt. Okay. Now, the best also is to save the whole session. So save it normally as a Chimera session file. And this is how we dock it. Now let's compare it with the original model. So what I will do, I will just recall the structure, the original structure. And you see this is kind of overlaid. Everything is in the same way because we haven't changed any structure from the original receptor file. So now when you take into account the active site and benzamidine, you can see that we came to pretty close results to the original model, original structure. So I think Chimera does excellent, AutoDock Vina does excellent job in predicting and docking your ligands to receptors. Now, if you are interested and you would like to design different ligands, you have to have molecular structures in PDB format. And what I can recommend is that you can do the following. To prepare a ligand coordination file, we have to know its structure. So we have to go to Tools, Structure, Editing, Build Structure. In this menu, we are going to build a fragment from SMILE string. And this will be the same, let's say, benzamidine residue. Okay. And to create coordinates, we have to convert the two-dimensional drawing of the ligand into three-dimensional coordinate file. So to do that, there are many tools you can do that. You can use ChemDraw, ChemDoodle, or Avogadro software, or you can go to PubChem and use this cute little PubChem sketcher to draw your structure and convert that into 3D format in Chimera. So let's draw the benzamidine residue. You click on here, start building your structure. Okay, this is this. I have to add a double bond here. And there is nitrogen. Nitrogen here, nitrogen here. And if you're not sure, you can also add hydrogens. This will be added automatically. And now you can see here, this is your SMILES code. So depiction of this chemical structure using this format. So let's copy that and insert that into our SMILES stringed file. Let's click Apply. Okay, let's see what is happening here. Oh, look at that. You already have it made in Chimera. So let's go File and save this. Save as PDB, and we can do receptor 2. Okay, we can give it a name. Okay, everything is legit. And that's how you create your ligands. To be even more precise, you can try to minimize the structure using molecular mechanics. So let's do that. Go to Structure Editing, Minimize Structure. Select that, and let's say do 1,000 steepest descent minimization steps and 1,000 of conjugate gradient steps. And let's see how this looks like. Okay, you will be taken to another menu. Now we're gonna add hydrogens. We are going to add gas Geiger charges. We select our force field here, and we are going to specify the charges. Yes, we have +1, and the structure is being minimized. Once you have it minimized, once we have it minimized, we can save this as coordinate file in PDB format. So receptor, let's say 3, and now we can use it to dock it using Vina. Once you get used to the interface and you have idea how docking works, I encourage you to go to AutoDock Vina website, where you can actually read and you can learn how to run docking in a script-like format, and you can use more advanced parameters using the software here, AutoDock Tools, that can prepare your more complicated ligands, and you have many more options to go after.